Hello friends, I always keep on saying that the physiology comes in every other subject of medicine. This MCQ is a classic example of that. This MCQ can be asked at the first prof level as a physiology MCQ. It could be asked as a pharmacology question. It could be asked as a FMT question because it's based on the toxins. It could be a medicine question, or it can be asked at NEET PG entrance exams. So let's see what the question is. Among the toxins mentioned below, which one binds with the SNARE proteins at the axon terminals? So it's very clear, it's toxins. That means it can be asked in FMT also, but the basis is SNARE proteins, which we learn in physiology. And the options are alpha bungarotoxin, or strychnine, or tetanus toxin, or curare. So got any idea which one could be the answer? Okay. Well, the answer is tetanus toxin. Now, let me say at the outset that all of these are toxins which interfere with the synaptic transmission. All of them, their action is that they interfere with synaptic transmission. Neuromuscular junction is also a synapse, and therefore some of them, they directly act at the neuromuscular junction. Others will act indirectly, but all of them have one thing in common, that they will affect the muscle contraction. Finally, they will affect the muscle contraction. Some of them will cause flaccid paralysis of the muscle, that is, the muscle cannot contract at all. The others will cause spastic paralysis of the muscles, that is, muscles will go into a sustained contraction, spasm, and therefore there is no movement possible, there is no muscle contraction possible. So that's common in all four of them, but the mechanism of action will differ. Now, before we discuss the mechanism, let's first quickly see what are SNARE proteins. SNARE proteins are the proteins which are involved in the synaptic transmission, whether it's a neuromuscular transmission or whether it's nerve-nerve junction transmission. SNARE proteins are involved. How and where they are involved? Look, there is neurotransmitter vesicle or neurotransmitter vesicles present at the presynaptic terminal. So when the neurotransmitter is to be released, whenever there has to be signaling from nerve to the nerve or nerve to muscle, neurotransmitter release occurs by exocytosis, right? And for the exocytosis to occur, the vesicle has to fuse with the presynaptic membrane, and then it opens and the neurotransmitter is released. In this fusion, the SNARE proteins are involved. They help in the formation of a bridge between the vesicle membrane and the presynaptic membrane. So this is where the SNARE proteins are involved, and therefore they help in neurotransmitter secretion, neurotransmitter release by exocytosis. And if a toxin acts on the SNARE proteins, then obviously it will interfere with the synaptic transmission, and therefore the answer was tetanus toxin. Now we will see where exactly those toxins exert their action. All right, you can see here that there is a neuromuscular junction shown here. The neuromuscular junction, this axon terminal has acetylcholine vesicles, and on the post-junctional membrane we have nicotinic acetylcholine receptor. We all know that, right? Now two toxins out of the four, that is bungarotoxin and curare, these two, they target the nicotinic acetylcholine receptor. They bind with this receptor, they target this receptor, and therefore they will prevent the action of acetylcholine at the neuromuscular junction. So what will be the result? No transmission of impulse will be possible. This will result in the paralysis of the muscle. So bungarotoxin and curare poison, they both, or let's say D-tubocurarine is a drug, they both target this nicotinic acetylcholine receptor on the muscle membrane. So that's the point number one. Of course, they are not interfering with the SNARE proteins, so that they are ruled out, curare and the bungaro toxin. So they are ruled out. That leaves us with strychnine and tetanus toxin. So let's see how these two toxins work. You can see here that this is a motor nerve terminal. You can see here, this is a motor nerve terminal ending on the muscle membrane, right? Now this motor neuron is coming from the spinal cord. So here is the spinal cord transverse section, and anterior horn of the gray matter has the nerve cell body of this motor neuron. So this is the anterior motor neuron. This is the nerve cell body. This is the axon which is coming to the muscle, and it is sending signal to the muscle for the muscle contraction. Now this motor neuron is innervated by some inhibitory interneurons in the spinal cord. If you can see here, there is a small inhibitory interneuron which is ending on the anterior motor neuron. So this inhibitory interneuron, whenever it fires, and it fires regularly, it will reduce the excitability of the anterior motor neuron. It will decrease the signaling sent by the anterior motor neuron to the muscle. Now, the tetanus toxin and strychnine poison, how do they act? They act by interfering with the synaptic transmission, well again, but at this synapse in the spinal cord, synapse between inhibitory interneuron, which is supposed to inhibit the anterior motor neuron going to the muscle. So this synaptic transmission is inhibited. You can see the diagram here. All right. So an inhibitory interneuron ending on the anterior motor neuron, which sends signal to the muscle. Now, how does the strychnine work? Strychnine interferes with the synaptic transmission at this level in the spinal cord. Strychnine is an antagonist of glycine. So let me put it here that glycine is an inhibitory neurotransmitter in the spinal cord. I repeat once again, the commonest inhibitory neurotransmitter in the spinal cord is glycine. In the brain it is GABA, but here it is glycine mainly. So strychnine poison acts like this. It binds with the glycine receptor on the postsynaptic membrane, and that is how it does not allow the glycine action to occur. Means this inhibitory interneuron will not be able to exert its effects on the alpha motor neuron, anterior motor neuron. So anterior motor neuron will fire excessively, and it will result in the excessive muscle contraction and spastic paralysis of the muscles. So that's the action of strychnine. Strychnine is the antagonist of glycine. It in a way it competes with glycine for binding with the receptors on this motor neuron, and therefore it prevents the action of glycine. If the glycine cannot act, if it cannot inhibit this motor neuron will fire excessively, and that the result will be spasmodic or spastic paralysis of the muscle. That's the diagram shown here. Glycine, and it is being released by the inhibitory interneuron, and then its receptor on the motor neuron. So this is where the strychnine will act. Now that brings us to the last one, tetanus toxin, which is the answer to this MCQ actually. Well, tetanus toxin also works here, but how? You must have heard that tetanus occurs generally after a dirty injury, a dirty wound. Yes, whenever there is such an injury, and this injury is deeper one, it has reached the muscle. So here is a muscle, as you can see. The tetanus or its toxin will enter through the wound, through the muscle, through the muscular wound. It will enter here, and then it will go in the reverse fashion. This is the motor neuron coming from the spinal cord. So tetanus toxin has entered through the wound in the muscle. Suppose this is some muscle. So tetanus toxin has entered. Now it crosses the synapse in the reverse, and then it travels in the reverse like this. It travels and it goes all the way to the spinal cord. From there, it enters this inhibitory synaptic terminal, this inhibitory interneuron. So tetanus toxin travels like this in a retrograde fashion. It reaches the spinal cord, and then finally what it does is that it prevents the release of neurotransmitter. Mostly it is glycine in the spinal cord. So this inhibitory interneuron will not be able to release glycine on the anterior motor neuron. Mark the difference between the action of strychnine and action of tetanus toxin. Strychnine was simply binding with the glycine receptor. It was not interfering with the release of glycine. Whereas tetanus toxin went all the way from the injury, muscle injury. It went into the motor neuron, went to the spinal cord in a retrograde fashion, and finally, as it enters this inhibitory interneuron, it will interfere with the SNARE proteins. It will inhibit the SNARE proteins in this inhibitory interneuron. Which means what? Which means that it won't be able to release the neurotransmitter glycine. So this is how tetanus toxin works, that it interferes with the release of inhibitory neurotransmitter in the spinal cord. And since there is no inhibition of the alpha motor neuron or anterior motor neuron, it will fire excessively. This motor neuron will fire excessively, and there will be spastic paralysis of the muscles. In fact, the symptoms like locked jaw or the other symptoms of tetanus and the respiratory failure, they all are because of the spastic paralysis of the muscles. So this was the mechanism of action of all those toxins mentioned, and we discussed all such interesting MCQs on our channel regularly. So it's a good time to subscribe to the channel and hit the bell icon.